Okay, good afternoon, everyone. We will start this afternoon's session. You've probably already figured it out by now. This is a beautiful room, and the acoustics are as terrible as this room is beautiful. So keep the ambient noise to a minimum, please, so that you can hear the great work that Wesley Clawson is about to tell you. Wes's current research involves studying neural cultures via a closed-loop system that I think he's going to show you a little about now. It allows for real-time interaction between a population of neurons in virtual worlds, investigating how neuron cultures learn and adapt as a collective intelligence. Wes.
Cool, thanks. Is this an appropriate volume? Okay, it sounds weird, so if I get too quiet or too loud, just give me a gesture. Okay, cool. So I'm Wesley Clawson. Like Josh kind of mentioned, I work inside the Weapon Lab in the cooler of the two buildings. So if you ever visit, you can find me there. And I'm talking about this. We'll alter the title, the tissue level intelligence via synthetic or computationally designed synthetic living construct. So the goal of the talk is I'm going to tell you a little bit, I'm not going to dive too deep into data analysis and some of the stuff we've done. I wanted to give a high-level idea of what the point was, some of the things we want to do with it, and where CD can enter into this framework. And the goal here is kind of regardless of the organism or thing that I want to interact with. For example, I'm going to talk about neurons in a dish, or a xenobot, or some very weird bio-hybrid thing that's made up of a swarm of robots, Twitter, and an ant colony. Is there a meaningful way that we can kind of come up with a library of tests of intelligence? I made a combination CDO. What now? Is it any good? Okay, it goes in a line. Great. That, where do we put that into our library of things? And once we have these things, as we go through, especially if once MomBot goes up and going, and we get more, we get a dad bot, a weird uncle bot, all these things can be automatically tested, and we can start coming up with catalogs of bioalgorithms, which is me taking algorithm and just putting bio in it to be cute. And then that loops back. What if at some point we can go through that catalog and say, I need to create something with this type of intelligence that uses this type of thought process, if I can use that very vaguely, and then we can make it. And so my job today is to get you to partially believe that this is something we can do. Because typically when we think about intelligence, at least I'm a neuroscientist by training, we think of a rat in a maze and it solves it, and we're like, yeah, so small. Rats do mazes, mice do too. They're just not so cute, and it takes longer. The goal, and I think I don't need to convince many of you here, thanks to Mike's talk, but also you're invited here as part of the group, is that any biological system could navigate something difficult, and you would maybe consider it intelligent in some way. It's not just kind of randomly bumbling around. And kind of the way that I think about this, I think is very nice. Jacob von Uexküll, I have no idea how to pronounce that correctly, defined this idea of an umwelt, which is like the self-centered world. And then someone wrote about it's constituted by more or less broad series of elements called carriers of significance or marks, which are the only things that are of interest to the animal. And it's not hard at all to take that definition and make it about any biological system and just cross out the word animal and put in like biological system. So you can imagine yourself like, oh, if I'm a cell, what are the markers of importance? Maybe it's pH or the electricity field around me. If I'm a genetic regulatory network, what are the things that matter to me? So this is how I started kind of thinking down this path. And so the cool parts about biology, we all know this, is that they can learn. So they can adapt and respond to things, and a variety of things. And what's absolutely cool is this happens across a variety of scales and spaces. So for example, I don't have a pointer, But, mice and can navigate mazes, but then whole tissues can organize their activity. Neural networks, whether they're synthetic or not, can do things. Genetic regulatory networks can learn, and then groups learn. There's group dynamics and group thing. So this learning, this adaptation, can occur across scales and spaces. Mike talked about this a bit. Here are some examples. 3D space, we're all used to navigating that. I can go get coffee without bonking into any of the tables most of the time, or it's a maze, but maybe something is navigated in transcriptional space when trying to reorganize how they can grow, or animals trying to grow into different morphospaces. Maybe there's a good morphospace and bad morphospace. How do we navigate that arbitrary space? Cool. And so intelligence, the way that I'm going to talk about it today, and kind of put forth the hypothesis that we can build this kind of library of tests, is that it's the degree of competency of navigating any space. So in a 3D space, I'm very good, but if you put me in a amorphous space, maybe I kind of suck at it. And so you have to go through and ask yourself, what are the different spaces that my system can potentially navigate? Given a goal, how can I achieve it? And I think this is not so buck wild. So again, Mike shared some examples from the lab where the GRNs can navigate this transcriptional space towards a certain end. There's a nice paper by Saram Biswas that he mentioned where they can do associative learning, conditioned learning, consolidation. Things can navigate physiological space, and I'll go over a specific example in a moment, where you have physiological adaptation, for example. Stealing Mike's example, if you put a planaria worm in the media with some barium, their heads degenerate, but they can adapt and grow resistant heads. It's pretty crazy. So there's some goal I would like to have a head, despite the barium, and they're able to reorganize themselves and achieve it. There's my goal. The normal way was blocked, but I was able to get around. I'm going to focus on the physiological space just because I like it and I'm most familiar with it. But you can imagine there's two phenotypic behaviors, purple and orange, or behavior 1 and 2, and there may be a host of parameters that kind of generate this behavior. So despite where I am in this three-dimensional parameter space, as long as I'm around here, my phenotypic behavior is the purple one, whereas maybe some perturbation pushes me over to orange. Despite where I am, I'm still orange. I think the most striking example of this is Eve Marder's work. She works on lobsters, and she's actually not far from here, Brandeis. And on the left, there's two pyloric rhythms. I don't want to explain what it is. It's a triphasic rhythm. There's a circuit. If it doesn't exist, they die. Okay, they use it to eat and read. And if I challenge you to tell the difference between the purple and the blue ones, you would find very little. There's 3 parts of the circuit. They're in a certain phase. You have the LP, the PY, and the AB. Because there's very little difference. Maybe the interspike interval is slightly different, but on the whole, the phenotypic behavior is the same. What's wild in their models, and later they do this in lobster, here you see purple and blue bars. Those are various parameters of the cells. So membrane conductances and synaptic conductances. And you don't have to be an expert at reading graphs to know that the purple and the blue bars are very, very different. So these are disparate circuit parameters that produce the exact same behavior. Just no circuitry has been changed, it's just the parameters of the cell. So for example, imagine if you had your phone, and if it was a little bit hotter, all of it reorganizes itself to work in a better way, given the heat, and then later it goes back. There's some pretty cool examples in Marder's work where they do this. If they take the vast parameter space of all things possible and then ask what's biologically plausible, the space is still big. And then if you say, okay, I'm going to restrict it, which of these parameter spaces are resistant to heat perturbations? Because the ocean gets hotter and colder, so it's something they're accustomed to dealing with. The parameter space that can deal with this is still quite large. And what's interesting is they might start here in parameter space, adjust due to the temperature, and when the temperature goes back down, they fall into a different space. They don't care. The behavior is the behavior. Okay? So this reorganization of circuit parameters is something we're working on in the system I'm going to talk about in a bit. More introduction. We talked about biological computation. Stephen and Thomas talked about it a bit. This is how I tend to think about it. It's not perfect, but it's based on Mars 3 levels. Typically, there's some kind of problem you're working towards, or some kind of goal. You need something to do the computation, and it's based on what they do, it typically produces the thing that you want. It's a bit simplistic, but it works. So for example, in the bird, should they want to fly, looking at its feathers is not enough. It's because it knows how to flap them in a particular way that it flies. And in neuroscience, if you go onto Twitter or Blue Sky or whatever you want, there's a great debate on whether these 3 levels are even useful, because they think, like, oh, we have neurons, they spike and you get cognition and consciousness. So, I don't know, that doesn't seem so helpful. But that's like taking this concept and only applying it to one specific tier of biological intelligence. What's interesting is every piece can scale. The way we tend to think is that I think, and Mike supports me, I think he thinks the same thing, is imagine, my GRNs, okay, are what the thing is doing. They have some kind of behavior that produces something that I want. I have a circuit inside of my head that's made up of neurons, astrocytes, glia, and what have you, doing a certain thing in order to produce a goal. These systems can kind of couple together. They work across scales, and all of a sudden, the scale of what the animal can achieve goes up as well. GRNs maybe can only navigate a very short temporal scale, for example, whereas I can plan basically what I'm going to eat tomorrow. After that, it's a bit sketchy though. So the goals can scale, the pieces can scale, the why can scale, the how can scale. And so if you put this dumb little 1, 2, 3 cartoon across these scales, it starts to become very interesting and helps put a little bit into perspective. If we're going to investigate this in a meaningful way, what do we look for? There's got to be something doing something. So in my case, I'm going to talk about neurons. They're going to be trying to solve a problem, and then the big question is like, what the heck are they doing? And this is nice, right? When these things can scale. Because in biological systems, there's a multi-scale consciousness. When I plan to give this talk, or when I plan to go to the bathroom and drive home later, I don't need to worry about what my lungs are doing. They can handle it. So these goals can kind of propagate upwards and downwards, because if I'm out of breath, my lungs take over, maybe some signals and like, don't forget to breathe. But sometimes I think about my breath, if you meditate, so the control can go both ways. And I like to do experiments and build stuff. So I like to think, if this can happen, if there's a what doing some things in order to achieve a goal, what are some of the mechanisms? What should we look for? What are signatures of this? Like something that I can look at. This is a wonderful paper by Paul Mason in 2015, going over, I think, conceptually some of these mechanisms. For example, if I wanted to write a sentence, I need a pencil to do so, or a pen. But if my pencil breaks, it would be very convenient to have multiple pencils on my person, because maybe if I don't write the sentence, something bad will happen. I lose my job. And many systems in biology do have this redundancy built in. Structurally, the pencils are very similar, therefore they can write sentences. Whereas degeneracy, like we saw in Eve Martyr's work, if I need to write a sentence and I don't have a pencil, but I can also use a pen or a crayon, or in an extreme case where someone murders me, I can use my blood to write on the wall. I can solve the problem of how do I write down the name in a variety of ways. So it's really interesting. Some systems, they abuse this redundancy, this degeneracy, but also pluripotentiality, just like cells. I have a pencil, for sure I can write a sentence, but should the need arise, I can also draw a smiley face, okay? Or I can draw the picture of the person who murdered me, or I can pin up my hair with a pencil when it was longer. Now, it doesn't seem to work. So some of the stuff that we look at in the system that I will describe here in a little bit is typically based in computation. What is doing something at the scale that I care about? How are they doing it? And the things that I typically look for are signs of redundancy, degeneracy, and pluripotentiality. And I don't have answers to all of these. I'm just kind of putting forth a framework that I think works for what we're doing. And like Mike mentioned, often in biological systems, the principles of multicellular plasticity, this learning, are obscured because things are involved. It's super complex. I don't know if you've ever looked at a neuroscience textbook is hard. It's like 3D, there's like a lot going on. It's really hard to investigate fully what a system is doing. But if we think that there is some kind of goal-seeking, problem-solving behavior, we may use that as a powerful invariant. As we investigate different CDOs, for example, if we take, for example, that there is this problem-solving behavior, it can help us analyze it as a set. Okay, so the analysis of building forms that have never existed before, such as CDOs, can reveal perhaps deep design principles as life as it could be. Okay? So if we can watch how goal-seeking behavior is achieved, what, how, and why, throughout many different systems, maybe we'll find some very deep underlying principles. And I won't go over this too much because Mike already really nicely described it, is when you have a system, you have to ask how trainable, how much can it learn, what can it do? And so Mike created this axis of persuadability in a very long but good paper called The Technological Approach to Mind Everywhere. You can see on the left, this is often the angle of the traditional neuroscience takes. So I'm going to go and modify specific genes or specific synapses in order to look or to cure something or to understand something. I want to go edit the hardware itself. Although there are some efforts being made to understand interval residuals, so maybe I can change the set point, the system will balance itself out, and I can watch how it does. The idea is, given a CDO, given a biological system, you have to guess where you are. Okay? And I'm going to start at C. My work starts here. I think that tissues are pretty intelligent. There's a collection of cells, many different cells, that can work towards a goal. And the general idea is I build a system that can train an artificial body. Okay? In your brain, it's hard to do. It's impossible to know what each part of the brain is trying to achieve at any given moment. So it's hard to put this framework in context. But if we can train artificial bodies or artificial goals, we can observe the what and how during and after training with the slew of tools available to us. Okay? So, how do we do this in practice, and why am I here talking about any of this? Again, we want to show that biological systems can be highly intelligent by solving different problems. Okay? And what's important is, if I'm a biological system, there needs to be these markers of significance. So I need to be able to perceive something that's important to me. I need to think about it and then act upon it. Luckily, other people have come up with a term for this called embodiment. So there's an environment, okay? And if I'm a cell, that's maybe the cells I'm connected to. It's my ECM. If I'm me, it's you guys. If we're CDO, maybe it's the government and companies. What is the environment of the organism? And it's acting upon us. We have a mind that at least partially processes the environmental signaling, and then a body or actuators or something that we can do either in order to change the environment or to kind of change our relationship to it. Okay? I'm going to be pretty non-specific with that. In general, that's the framework of the body. I try to make it as simple as possible because I'm a little lazy and also brains are really hard. So we virtualize every piece of it except for the mind. So we're going to talk about a virtual environment and kind of a virtual body. Through this closed-loop platform. So here, I'm going to talk about how we grow cells in a dish. Right now, we interact with it electrically, but we're working on chemically and optically as well. Basically, we're going to take the output of a neural culture, and you can define that to be whatever you feel like. Decide if it's the thing that you want or not. Okay, and apply a punishment, reward, and throughout the whole thing you measure. Okay? So I can create virtual environments by stimulating, perturbing the system in a variety of ways. In the same way that you see stuff, electrical signals eventually hit you. Okay, so I control the entire environment of the tissue. Okay? And I can also control how I read out what the tissue does and how that affects the environment. That's the virtualization of the body there. So how do I do this? It's with MEAs, microelectrode arrays. It's pretty cool. In the past, they were big and fat and very hard to do anything. But thankfully, other smart electrical engineers created CMOS electrodes. So now they're very tiny and very close together. So the devices I'm going to show you, there's 26,400 electrodes and roughly 8 millimeters squared. But I can record from a large number of them with extremely low noise, up to 2 to 3 microvolts, at 20,000 samples a second. So it's a lot of data. And just a little primer for non-neuro people, cells are electric. So when the neuron fires an action potential, it's kind of hard to see the colors, sorry. There's typically negative deflections when it's extracellular. And then the other parts of the cell will be positive. So you have an axon where the thing is going to go, and the dendrites that are picking up. So these MEAs report extracellular local field potential. Hopefully you can see this if you're on the TV. Here is a graphical example of what it looks like if you're looking at 4 neurons, 4 different colors. And I'm looking at the waveforms that were produced by a spike neuron. We'll look at them more closely, but here each little square is 17.5 micrometers. So it's a pretty tight little patch square. But I'm looking at single neurons. It's pretty cool. I can zoom in. I'm very clever with how I pick the electrodes. And here I'm looking at the electrical footprint of the soma because the deflection is negative. And the dendrites are hanging out down here because when it fires, I get positive deflections here because of the way the current travels. What is that dendrite going out to? Well, it turns out there's a collection of neurons here, and that dendrite is touching. So I can start. I can use microscopy for sure, but it's hard because these chips are opaque. But I can look at the electrical footprint of these tissues. Here's the cool video that I brought to show you that. Here you're seeing the action potential, fire, and literally it traveling along its axons. So these electrodes are so close together, they're so sensitive that I can actually watch the action potential move along the axon.
This is for one cell?
So this is one cell, this is another cell. Yeah. Now there are many cells around it, I've just isolated 2 key units. So here the action potential start. You can see the different colors as they move, that's electrical activity. So here I'll go through the game because it's relatively short. It fires in a minute. And then it's traveling down the primary axon here. And the red lines is just a computer being really sure. What's nice is, we could watch these all day, I have many of them, but it lets you kind of electrically image the neurons where they are. And because you can do this, you can ask questions like how fast did it go, how far did it go, where did it go, who is it likely to have touched on the way there. It's pretty cool.
I haven't heard imaging this percentage of electric current move through the.
Yep. So it's
a proxy.
No, it's literally that. It's the local field. It's important to note I didn't invent this. Okay. I use it heavily and I've added to the code base. So this is part of the product of this Maxwell Biosystems created. I added to it and it helped out with a couple things. Unfortunately, this isn't in real time. You have to record it enough to build like a model of where you think it is. But it lets us understand functionally how the connections work neuron to neuron in the dish. So you can imagine recording this a bunch, checking it all out, doing some kind of training algorithm over many days, and then looking afterwards and saying, how is it different? And if we do this 1,000 times, does it change in the same way? What type of algorithms are they following in order to change how they're communicating? Because most neuroscience will just say it's synaptic, it's in the connection, and move on. But there's a lot of things going on. It's not purely synaptic, they can myelinate. Sometimes, even if we're heavily connected, we might not always send the same signal every single time, and this lets us investigate. Great. Just to be clear, the way that I do this is we take mouse or rat cortex, and similarly with EC cells, you dissociate them, so you take out all the cells from the ECM. This includes the astrocytes, the glia, the other things like that. You strain them, you make them very nice, and you plop them on a plate, and you keep them happy by feeding them occasionally, and they will grow into a tissue. For example, here's a nice one where you can see single neurons growing in the MEA. Sometimes they grow into clusters. Working on that. And then here's the typical data that the machine can see or that you can see afterwards. So here's like the 1,000 channels that I happen to record in this experiment. And every time there's a dot, the machine is recording that it's pretty sure there's a spike there. So these are spiking neurons. And if I was trying to sum all these together, that's just spotting things. And you can see here there's 3 big peaks plus a little peak. Those are called bursts. They spontaneously occur. I'm not stimulating anything here, they just happen. They happen in you, some too. If they happen enough and they're very bad, it's called epilepsy. And the hypothesis is that they serve some kind of function in the culture to maybe help spread connections or establish some kind of tissue-wide communication. The hypothesis is, it's still a hypothesis, nobody really knows. But what's nice, if you're going to build a closed-loop system, something like bursts, they're relatively slow. They're on the scale of seconds.
You're operating channel space, right? You're not spike sorting?
Here I am not spike sorting, but it's channel space. But I do cut out redundant ones and stuff like this. But no spike sorting. Cool. So cultures, these bursts are slow, they're easy to see, and they occur across the whole tissue. So part of our early training requirements is I'd like to train the tissue, not specific neurons. I don't want to go stimulate two neurons and try to cause synapses to change. I want to give a goal to the tissue. I'm not very clever, but the tissue is very clever at how to manage itself. I'd like to understand how the tissue decides to alter itself in order to achieve this goal.
This has any connection to the default mode network, the baseline?
Kinda. Like a default mode network, kinda. Yeah, exactly that. So if you do default mode network, if you sit in a chair and do nothing, that's the default mode network. And neuroscience researchers will do like fMRI or EEG when you read the default networks. Just as a note, the default network is very dynamic. It's not stable. Cool. I'm also not the first human being to think about training cultures, just to be clear. They go back to the 90s, so Shahaf and Iran, great work. The most famous probably is Steve Potter. And then most recently, this relatively controversial paper from Cortical Labs where they tried to get the neurons to play Pong using a similar system. Sort of worked. So here's the team that helped me originally build this. Many of them, Trevor's at Google. Nathan actually works for the company that makes our system temporarily. But they helped me go from kind of the physical world to the virtual. And to talk about this, the system that we built will be freely available to use should you have the system. Because we wanted to train the whole tissue, so holistic. We think that it's agential, okay, so it's a collection of neurons. The neurons themselves, I think, are agential. But then the tissue is a collection of cells, is also agential. And the point was to look at learning, to test intelligence. Can you learn? How can you do these things, and where does it go? And that very conveniently spelled out how. So this is our how system. It also happened to be the name of the man that funded me for this. So thank you, Hal, if you ever hear this. And we can use this system to go from here, for example, this is a burst that started on the right of the chip. This is the MEA in plot space. And you can see most of the neurons start firing over here, and it went to the left. Roughly. Okay? And then here, there's one that starts roughly on the left, and at the bottom part, it goes to the right. The directionality is not that important, it's where it starts that's important. So here I'm looking at this one was left, this one was right. When you look at those two graphs, they might look the same to you, but they're different. And so our virtual environment, we started very simply. There are two places to be, on the left and on the right. It's a 1D environment with two spaces in it. And we thought, okay, one space will be in the environment that has some kind of bad negative signal. We chose a random 5 hertz signal, so it's 5 hertz. But when these pulses come within the 1 or 5 seconds, it's random. And then the other space in our virtual world has no simulation in this example. Okay, so it's kind of dumb. If it started on the left and went to the right, we moved the virtual regions to the right, and we got a random simulation that we think is negative. If it started on the left and moved to the right, we moved it into the no stimulation. So here is an agent, a mind. These are actual cortical neurons sitting in a dish. Their behavior, these bursts, cause a virtual agent to move into an environmental space that has very strong, we believe, negative feedback, and we can talk about that in a minute. The other behavior that it can do puts it into a different environmental space where there is no feedback. And the hypothesis was that they would eventually only exhibit the bursting that put them in the no stimulation box. And this is a nice graph for this is the middle of training. So before, it was about 60% on the side that we wanted. And we switch back and forth sometimes, so not biasing. So proportion of success. So it successfully started on the side that we want. Already it's 60%, but after training, it went up to about 90%. Of course, we did this many times over many days. So this top graph is a nice example of this, where we had 5 or 6 different cultures. Here's the proportion, ignore the title, sorry, of successful, the proportion of the side that we were training for. And in general, they go up, which is very nice. Meaning that over days, and now we've done more analysis, just a quick graph to see the proportion of diversity on the side that we wanted was up. Just to double check, these are some cultures, 2 of which got stimulated a lot for a different protocol entirely, and then 4 got nothing. They just lived their lives in an empty path. And that proportion doesn't change. We've now done this many times, but if you look at too many lines on this, it's kind of confusing. So we think we're able to cause some tissue-wide change. So we're able to reorganize and burst selectively from one side of the tissue versus the other. And again, now we've done where the reward is a regular stimulation. We've switched up, we've done a lot of different conditions. I just wanted to show that it's pretty easy. Other things that we are doing in a similar vein, this HAL system now, you can create any virtual world you wish with any environmental feedback. So some students have used, for example, how do we create virtual movement from a tissue or from a CDO, should it be a CDO. They broke down the data, they used a computer with training data to build a PCA model, and then during the recording, they were able to translate that into virtual movement. So where you are moving in PCA space, you have your virtual movement. Now we're testing that we should be here. What happens when the goal changes? So a big problem with machine learning is overtraining. Once it gets good at one thing, it typically sucks at another. So if the goal changes, the machine learning models can fail sometimes. So is biology better at this? If I shift the goal regularly, will it start to predict the goal change? That's another form of intelligence, statistical planning. Can we do this at a state-based level? So there's ways to classify your data into different states, for example, A, B, C, D. And maybe one state you've classified with maybe something like Thomas Darwin does, or maybe you have a tissue that has a disease, and state B is associated with phenotype that you don't like, for example, a seizure. Rather than going in and trying to fine-tune all the connections, or blast away one's other tissue, or give a bunch of drugs, it's destructive, can we train the tissue to stop creating the dynamics that are associated with seizures before they start? Then, of course, right now, all of our interactions are electrical, but there's also what we've been working on, time-based chemical stimulation. If you've ever worked with fluidics, that's awful, so it's just taking time. And soon we'll be adding, one day, optical, where you can maybe uncage specific drugs, or you can optically stimulate something and record optically at the same time that you're doing this electrical thing. It's quite cool. Something I'm really jazzed about, I'm currently briefly funded by DARPA. Here's what one of the dishes looks like. There's 6. And one, it's public with DARPA, but 2, I don't mind telling you because I'm the only human being that has the code and the capability to connect these wells virtually. So we can have one culture, its environment is signaling from other cultures. Its actions can be translated into signaling that then is delivered to other cultures. So now we're going from a collective of neurons into a tissue to a collective of collectives. And we can modulate this communication scheme. We can also modulate what's in the dish. When we put this or that together, what's better for certain tasks? If we had these tests of intelligence, maybe when I do 2 cortical pieces, 2 hippocampal pieces, and 2 thalamus pieces and connected in the following ways, that's way more intelligent than if I had just used 6 cortex. Or it's more intelligent in this way, but in this way it sucks. So slowly we're building these virtual worlds that can test different types of intelligences, as well as kind of create these cognitive meta-agents that are collective of tissues. And of course, there's tons of questions similar to the things that Thomas has asked about dependent upon how you connect them, how is a learned behavior stored inside of one culture? When we start connecting them together, how is this experience spread across the world? It's nice. I'm not in the brain where it's confusing. I need to control everything. So I do know how it's like, I know exactly how they communicate. They must, because they're physically separated. But with great power comes great responsibility.
Recently there has been a lot of work on memories and information being stored in non-neural cells, right? So could you do something like that?
Yeah.
You would have neurons and astrocytes and...
Yeah, so keep in mind in any of these at the moment, there's also astrocytes and glial cells.
Oh, they are?
Yeah. So there's some cool papers that show that astrocytes support maybe how memory is stored in the brain because they regulate energy essentially. So now, the hard part about the brain, it's 3D, and you can never know where things are happening. Right? It's just impossible. Don't let this neuroscientist fool you. We don't know how the brain works at all. It is a gigantic mystery. And I got fed up with it, and it was like, I called Mike randomly one day before he hired me and said, what if it was 2D? What if we used all your cool bioelectricity stuff and we did this? Because it's like 2D. Soon we'll have, we can grow the tissues only in the sensing area, and it will be if we can find some, it would be the world's first fully recorded complex biological system doing a task. I mean, imagine if you're a theoretician, or someone that wants to test a theory, you can fully record all the bioelectricity in the dish and do infinite perturbations as you wish in order to better understand how it propagates through a system. Just keep in mind, they're redundant. They have degeneracy and pluripotentiality, so you can do these things many times to pull out the way that they are thinking or doing these computations. So the last couple of minutes, because I have a couple of minutes though, right?
Yes.
Okay, cool. Is how do we bring CDOs and HAL together? And there's two kind of axes, in my opinion. Is often, like right now, what I just showed you, I wanted to train the Y so that I can understand the neurons and how they, or the astrocytes and the glia do. So I trained an artificial Y so that I can explore the what and how. Given a new CDO, like a xenobot or a different form, give the Y to explain the what and how a little bit. But similarly, we can say, given a what and how, what Y's can I attack? And is it better than some what and how? So it kind of informs how we are CDing the organism. So for example, you can ask an AI system that is parsing through all of these data, microscopic data, LFP data, training data over days. It's terabytes of data, it's a little intimidating, but like what perturbations tend to produce the phenotype XYZ? If I train with signal A, is it better than signal B? Or what should I take, what components in the dish would be better or worse for a given Y? And then, of course, we would try to understand why is that. How best to combine neural behavior and tissue data at the same time in order to control a robot that's running around serving coffee? I mean, it gets to the question, there's now a non-trivial number of startups trying to use neurons to compete. In my opinion, they're all doing them relatively poorly. It's an interesting line of thought. And what's interesting is, given this data, multiple tissues, multiple tests of intelligence, all of this data, microscopy, you could do RNA sequence. I mean, think about it experimentally. If Mike or Holly showed you anything, you could do any of that here. You just need time and money, unless you have like an Uncle Bot that does it. Can we start to pick out like general principles of how? Okay, every time we look at neurons doing a certain task this way, they seem to reorganize following this set of like internal logic. And then can we take that and better decide with machines? We still are pretty limited computationally. And there's a big question of like how can our brains can do so much more than a computer? And teasing that out is very, very difficult. But it's a good task for AI, I think, because of the scale of the data. So how do we integrate computationally designed organisms in a loop like this is you do it in every piece. You can have a computationally designed organism. Soon, I can only come up with so many tests of intelligence, or us if we're sat here for a whole week. But maybe the algorithm, maybe the AI, can have computationally designed tests of intelligence to to try to pick apart something very specific. Maybe you can start thinking out these kind of bio algorithms using something like these artificial intelligence that used as well. And then start to inform how we create new computationally designed organisms. If we know we want one that can do a specific task, you go into the library of tests, these bio algorithms to say like, if you stick neurons with one area, you blend them up like this, you might start to see this, because the way they work will probably work on a time scale that's possible for you. So yeah, that's what I do in my group. I hope that was not absolutely bananas. I want to say thank you to Mike and to Josh for letting me be here, and Mike for having me. And my team members, thanks for that also, Josh. Taylor Grasso, who just finished her PhD here at UVM. And Sydney Palmer and Serena Dean, who helped me run the wet lab side of stuff. Past team members, Nathan, who was going to grad school at Harvard in the fall, Trevor, a little slow for me, Viraj, who was doing what does what, and Ed, it's about the university. He helped us in high school. Plus all the other 11 lab members. I was not a biologist at all when I started, but because of Devin, Juanita, Hamid, Axel, everyone listed, I could actually do tissue culture and not destroy it. So thanks. Yeah.
Any other questions?
Yeah.
Question, I'm sorry, probably have trouble conceptually to understand some of the points that you did. But how, so for example, so essentially you have this two-dimensional tissue of neurons and things like that, right? What do you think it gave you more than, first of all, how many cells were there?
Sure, yeah. Here, there's roughly 150,000 cells, and a third of them are neurons.
Right. So, let's say 50,000 neurons, okay? So, how did you test it against actually a purely synthetic neural network model? On the same, if you were to retrain on the same training set that you have, how different would it be actually from?
Yeah, no, a machine would outperform it like a million times.
Right. But what do you think then is the actual, in this model, what does the biology give you more in this context? I mean, this is, I'm sorry.
No, it's a phenomenal question. I can talk about it some. And if I'll answer it briefly, and then we can jump after the fact. Because this is the problem that many of these startups are facing, because using biology as compute is a very sexy idea, because like brain work real good. But then what does it do better than a machine? Nothing yet. There's a really great study, it's a group in Japan, and I think it's the Takahashi group. They trained neurons to solve, or try to solve a little bit of the cardboard problem. They actually recorded data and used it to build a model that solved the cardboard problem. And 100 out of 100 times, a basic machine could beat it every single time. So the conclusion of the paper is like, why did we even put biology in this? It just made it worse. So to answer your question, I think there are algorithms inside of you that you can pull out to make a better machine. Or I think, that's because I think the way computing is done now, machines can be improved with these things. But also, I think in terms of things like generalizability, I'm not overly specified. I can do many things-ish. And so I think the big task, and I don't want to give my opinion away, but is what can they do better than the machine? Because whoever can answer that question in a clever way and make a company out of it,
and I completely agree, and actually, because I did play that before, this concept of at every level we might model the system as an autonomous agent. That has an environment, so it has sensors, it has actuators, but it also has some processing power. The question is really, at each level of those listed levels of complexity, maybe one of useful things would be to understand what those things sense.
Yeah.
and how they actuate, and then maybe derive some of the embedded logic through trial and error and through learning from
Yeah.
its outputs.
That's the exact path we're taking with
And
some of
then how
this.
they nest.
Yeah.
I mean, that is really, I think, probably the interesting part.
Yeah, I'll quickly answer and pass it along. Because we have two levels at the moment. You have neurons and tissue, and so you can actually investigate quite heavily what a neuron, like the agent of that is the neuron, but also how it's participating in the collective that is the tissue. Now that we're doing these 6-well things, you have another level, this third level. So we can really start digging in quite deeply about how these levels are interacting with each other. And I think that's when we're going to start finding stuff that's more special than typical. Briefly. Did you have something to interject or just a question?
Well, I was just going to say, another answer for your question would be, the brain is vastly more energetically efficient.
Yeah.
You know, any cell-top computer, you can like run the human brain on Apple, where you need a lot of power to run the laptop. So if we could figure out how to harness the neurons or to extract the powers that the neurons do, you could do all of this for pennies on the computational dollar, just because it's evolution has made it much more.
Yeah, you either have to be better, faster, or cheaper, and I think cheaper is the easiest of those axes to think about.
But not for every problem.
No, I agree.
That really is problem dependent. I mean, you see how much energy you use computing, you know, the thousands of digits of pi. Right? So, I mean, it's all relative to which problem is adapted to the system that computes.
Yeah, and that's why this why piece of that why, what, and how, or why, how, and what is so crucially important because they're interlinked very intimately. Yeah.
2 questions. The first short one is why 5 hertz is a random standard?
Sure, yeah.
And then 2, like at the beginning, Mike had posted like less Skinner, more Montessori. This is all Skinner, so I'm curious, like, how you're thinking of maybe, what learning means.
Yeah, I mean, I work for Mike, but they're still not necessarily fully aligned. No, but and also sometimes he does stuff and it takes a while for it to propagate down to me. So the way that I talk about this in a year may be more aligned with what he presented this morning. So that's the kind of the but some of this, the framework that I presented comes from how I thought about stuff pre-Mike. So maybe eventually we'll mix it to some collective. So then the 5 is relatively arbitrary. It's a decent hertz. It's generating a lot. It's near theta, things like that. Why random? I really wanted to test this idea based on Carl Friston's free energy principle, where if there's some underlying principle that biology does like to reduce uncertainty in the environment, I thought the worst case scenario is something random, comma, that they can still sense. If it's too fast, they can't tell. If it's too slow, they can't tell. So we picked 5 hertz because it's a good hertz for a neuron, and randomness because we got random to that. And then we're doing trials. Put the data up here where for a reward we do something irregular, or we take it away to make sure it's not just the electricity and things like that.
So piggyback off of Eugene and Gina's questions, I'm wondering how much you see the scaling of this technology broadly, everything that you and I do, as something that starts with the cell and then grows out from there. And so maybe in the future, if we're designing larger scale organisms, we impose some large biological field or something, cell organisms, versus how much you think it might factor through larger modules that we create. Right? So like for the Skinner thing, what if one of the things we look for is can we train a 10,000 cell xenobot that learns how to learn, right? And so you can give it higher level signals that propagate efficiently for the organism, and then maybe even larger things from these modules.
Yeah, I think that is a more realistic thing. Is because also the idea of hand designing a circuit seems like a nightmare. I mean, I have a master's in electrical engineering, and it's hard. That stuff is just difficult. So it was very appealing to me when it's like, this multi-scale competency. If I can find an appropriate level where I can give a gentle or a strong control signal, and you will self-organize, that's perfect. And then we can start using the 6-well thing to test about how to do this. Is exactly kind of the idea that I had is you have these two independent electric systems, but if you let them talk, if one learn, not to give away the next experiments, but if you have a trained one and an untrained one and you let them talk, how does that propagate potentially, and how do they start connecting, and can we find clever ways to connect them such that maybe it's like a sourdough starter, where I never quite, the learning is always somewhere, and as you keep adding stuff to it, it just keeps propagating. And how that interacts with whole organisms is super, super interesting. Sorry, like 3 people raised their hands at once, and you're the closest, so. Sure.
Yeah, have you seen whether, whenever neurons or other specialized cells can undergo more complex behavior based upon the environment, they somehow de-differentiate before starting a new journey. Right, like they need to erase part of their previous memory to acquire a new context and then go further. Because this may be an interesting phenomenon. Whenever apparently in vivo, whenever there is a population of cells that is within the context of the multicellular environment, and a new tasking is imposed, for instance, in the disease state, you always see that some of the cells they differentiate to acquire more wider capabilities, and in that particular moment, some mistake may occur, especially at the level of those more recent biological systems in terms of evolution. For instance, if you take seven-helices receptors, it's like saying that you have multiple components, multiple target stuff, and so the error can be much better controlled. While if you go to 7-3 kinase receptor, it's just one component, multiple targets, so one single error will affect many downstream cascades. And it's a matter of fidelity going backward and like in time, the evolution of time, recapitulating this package in a very fast way before. So this may be.
That's super interesting because we didn't talk about lifetimes, but these cultures come from embryonic animals, and they typically die around 60, 70 days. Sometimes they'll live to 100, but iPSC cells can live for years and years and years. So iPSCs, yeah.
Inducible pluripotent.
Yes.
There is a huge problem there.
Good to know before I do it.
For instance, they've been obtained by stable transduction in the receptor stability states. Now, those genes, they encode for molecular motors that are supposed to stretch the chromatin and allow more space for larger, in terms of dimension, transcription factors that are tissue specific. So there is a magic moment in embryogenesis where these genes, they first go up, and the cell is not undifferentiated anymore, it's simply pre-permitted. Then these genes need to be shut down because this is a magic molecular biology where the genes go down in transcription, the peptide product decreases as well, and the chromatin is still stretched, allowing more space for the tissue restricted transcription factor that are already expressed at the very basal level. So once they get in, they do feed-forward stimulation with their own gene expression. If there is no biphasic phenomenon that could not occur in IPS because there is stable transduction that makes the somatic cell becoming embryonic-like. But as you know, the yield, the differentiating yield is tremendously low. And this, it's not a matter of quantity, it's a matter of quality. So if you translate this in terms of your fantastic concept, we need to move from quantum to quality to order of units. It's like you just driving a car with potential engine of Ferrari with the brakes. And then
a fun ride, but dangerous one.
That's important.
I'll have to look into that, because if you have cells that can live a long time, then you can do some of the experiments you're talking about, or perhaps they reprogram. Because it's on the docket, if someone has aging funding, can new cells learn something, can old cells learn something different? How much can you learn before you collapse?
It can be useful to obtain IPS that have been created with non-viral vector-mediated gene transfer technology. It's more tough, but
It's
At
possible.
the end of the day, it's possible. At the end of the day, you may have.
Maybe Mike has a connection. He hasn't.
I wanted to, this is more of a question than for everyone, more of a philosophical question maybe to Mike and to Josh as well. In the sense, what are we trying to achieve with the new sort of concepts of biological computation? In the sense, you can look at it from two sides. You can create organisms to compute something for you, right? So they react to some stimuli and they give some output. But it might be, and this maybe comes closer to what Mike is thinking, that in biology the computational media is in some sense the organism itself.
Yeah.
So maybe the solution to a biology computation problem is the organism itself. So it's maybe something that, can reframe a problem to solve in such a way that the solution is to grow an organism that satisfies certain constraints or certain things. Like what Carl was saying is that in biology, as cells themselves somehow find change their phenotype in function of the problem that they need to solve to survive or to thrive or whatever it is, So maybe the view should be not only to utilize organisms to compute for you, but also to be.
Yeah.
And I don't know what your thoughts are about that.
I mean, just as a quick comment, and it's a comment for everybody. But training artificial wise might not necessarily be, oh, I need you to compute something for me, but I need an organism that can handle this task. I need a postdoc that can do this, and so I grow them up the way. So it's not necessary. When I say compute, it's not necessarily like I need an organism that can compute 2 + 2, but I need an organism that can be, that solves these certain set of tasks in certain ways. So.
Or even I think the shape of the organism is the solution to a problem.
Yeah. Like
for example, in physics, you have shapes that are solutions to reunification problems. Where you probably have some sort of reunification problem in some, atomic space or whatever it is that constrains somehow. But the constraint defines the solution to some degree. Maybe that's one way to explore. I mean, it's much more complicated.
But yeah,
of course, but it might be that it's worth thinking even in some toy models.
Yeah, so we have something like this that we've been trying. We have something like this that we've been trying to get off the ground, which is to basically train tissues for specific morphogenetic outcomes. So it's basically what you just said. You set up a set of stimuli and constraints where the solution would be to grow the way that we want them to grow. There's positive negative reinforcement and do certain things. So this is, there are technological challenges, but it should be solvable. It's an engineering problem.
Just very briefly, I think we're trying to attack a lot of assumptions in a lot of different fields. One of them is Cartesian thinking, right? So your comment, should it do or should it compute? With respect, that's very Cartesian, right? So from life's perspective, being and computing, what's the difference? That they aren't fundamentally different things. So the blue and the orange here, like who cares? Maybe we somehow ask an anthrobot to solve a problem and it quote unquote says, I don't know how to, but I'm going to go I'm a collective and I'm going to recruit some other collectives and we're going to go do stuff together and the result will be something that you as a Cartesian person will view as a compute, but from our perspective, it's just a thing. It's just life. Yeah. So I think just by doing the work that Wes is doing, all of this work is attacking this Cartesian viewpoint.
Yeah, and I think what's interesting, I didn't mention it here, maybe I should have, but if they can't solve that problem, an interesting question is how do you map the problem space that you have to a problem space that the biology can do? And when you can do things like that, that's when it becomes extremely powerful.
That's what I'm saying about the sensors and actuators. That was the problem in robotics. That what robot senses is not what you think or see that they sense. It's completely, they have a completely different perception of reality. And that's why it's very difficult to, I mean, that's why, by the way, the sort of the top-down approach in artificial intelligence didn't work for 50 years, because it was too complicated to create the representation of the environment and of the space as we see it translated into the robot's mind. But the second is, if we have this different nested competencies on, at least we have this, to understand this basic interface of this thin client, which might be saying in between each level. And why the levels that are above somehow symbolically constrain what is below. Because if they weren't, maybe the below would not exist. Right. I think it's almost like a logical principle.
I didn't dive too deep into the analysis, but that's typically how we analyze some of these things, is through symbolizing and looking at kind of computational properties based on higher order signal. I don't know, I think I'm like way over time.
Any last question? One more question.
I will fail during the tea break, so we can argue about this stuff too.
I'm going away.
Yeah, okay.
Have you looked, so you're looking mostly at the global bursts, right?
Yeah.
The very global summary.
For this example, yes.
Have you looked at things like, instantaneous channel correlation? Like, are there other ways you can compose the power activity?
Yeah, I made a stupid joke with great power comes great responsibility. The system works, it is quite good, and Caitlin is just making it better. And now you could theoretically do anything that you can measure in a reasonable amount of time. So we thought about training for like time-windowed synchrony or correlation. I mean, you could do anything. We have some preliminary trials that work pretty well where the tissue, if you deliver a frequency, like a resting heartbeat almost for the spiking, they will they can raise and lower their firing rate as not the bursting, just like the quiescent stuff. You can get them to raise and lower it a little bit. I'm trying to tinker around. This is the hard part, is that you have to do all this by hand. So with the tinkering, you see, like, can you get them to like raise their resting heartbeat, or the next thing we want to do is try to shorten the interburst interval or make it longer. But yeah, any of those things are definitely possible. The internal logic of the system is just like, look at this, if that, then this. It's pretty simple. Yeah, you're limited by what you can measure very quickly, because if it takes like an hour, then it's too late to do anything, I think. Again, I think the time scale that you interact with the CDO, regardless of what it is, biological systems typically have intrinsic time scales that they can deal with. Maybe we can talk about it as cognitive light cone or however verbiage you want to use. So if your task does not fit within the dynamical kind of window, then it won't do anything.
Okay, we reached the middle of the afternoon, and again, thanks to my terrible organization skills, there is no coffee and pastries available. So if you're feeling the need for caffeine or a soda or even some fresh air, you can head outside. If you need caffeine, put into your phone Henderson's Cafe. It's here on campus. It's a 5-minute walk. You can go get a coffee and come back. Henderson's Cafe. We're going to take a 4-minute break, and we're going to start up again exactly at 2:30 with Manish Patel. Henderson's is not that fast. Henderson's is not that fast.
I'm curious as how.
Hello. Let me just get things set up. Let me get set up here and we'll do 2 minutes. Just get set up.